great lunch. This has been action-packed thus far, so we're really encouraged by the discussions, the learnings, and the continued passion around all the stuff that we're doing. So I'd like to introduce this panel. We call it the oncology panel, but I think what we really have here are a lot of drug discovery and I'll call them biomarker specialists. Some of them have been in the field for 10, 20 years, and proteomics was one area that started in a lot of electrophoresis and moved into mass spectrometry, and I think now there's evolutions into ELISAs and digital ELISAs. And so I'm going to ask each of the panelists to introduce themselves, also introduce their work and some of the areas that they're currently focused on and some of the intriguing paths that they're trying to take. And with that, then Thomas will probably end up helping me moderate toward the end. So maybe we'll start with you, Thomas, and we'll go from there.
Hello everybody, my name is Thomas Joos. I'm a biochemist by training, and I took over the NMI biochemistry department in '98, focusing right from the beginning on multiplexed immunoassay technologies. And over the last 2 decades, we are now more than 35 scientists, technicians, engineers working on biomarker assay development, sample screening, and we are one of the rare academic institutes who are actually running clinical trial samples.
So hello, my name is Rudolf Oehler. I am from Vienna, from the Medical University of Vienna. I am working at the Department of Surgery, but I am a molecular biologist myself by education. And we are working in the field of breast cancer and colorectal cancer. I'm mainly interested in the interplay between these cancers and the immune system, and look for biomarkers to describe this interaction and how it can be used to predict the response to therapy or to help the clinician to decide which therapy should be taken.
Yeah, my name is Barry Vanderstrate. I'm the Director of Bioanalytical Sciences for immunoassays, and I work at PRA Health Sciences. So I'm one of those people who work at a CRO. Within the science department, we are responsible for developing, transferring, and validating methods, all kinds of immunoassays. So that's for PK, that's for PD, immunogenicity, flow cytometry, but also biomarkers. Yep.
Hello, my name is Stubenrauch from Roche, Roche Diagnostics in Penzberg, close to Munich. I'm a biochemist by training. Currently, I'm heading the bioanalytical science group in large molecule bioanalytics, which means together with my labs, we take care on PK assay development, immunogenicity testing, and as well as some kinds of protein biomarker assays are also developed by us.
Hi, I'm Darragh Duffy, an immunologist working at Insitut Pasteur in Paris. I joined Pasteur 7 years ago after working mostly in vaccine development, and originally joined Pasteur to work on the biomarker field in viral hepatitis. And there we had a nice approach for combining a genetic SNP in IL-28B and plasma levels of IP10, so combining genetic predisposition plus a protein circulating marker that was closer to the disease relevance. And this was a really nice approach until the drug companies came out with much better therapies that directly target the virus. So it made our biomarker strategy completely redundant. But like any good scientist or innovator, we pivoted and we've applied this more general approach to understanding healthy immune responses. So combining genetic associations with environmental perturbations and trying to understand what really shapes a variable immune response. And the second direction we went in was to discover the Simoa technology, really wanting to push the sensitivity of measuring circulating cytokines. So doing a lot of work in type 1 and type 2 interferons, and that's opened up a whole new area of not just infection, but also autoimmunity. So my 2 disclaimers for this panel is I'm not at all an oncologist or even an immuno-oncologist. And as an Irishman living in France, I don't at all understand the American healthcare system, so don't look to me for that.
Got it, Darragh. And I'm a Slovak trying to survive over in the United States. So I may want to start with you, Darragh, but maybe go back across and then Thomas will take it from there. But I'm real interested if you could describe specifically the role of sensitivity and what you think maybe you haven't uncovered yet with the sensitivity levels that are currently available that you think might be just a doorway away. And then if we could go another 100-fold in sensitivity, would that, in your mind, further help your efforts in the level of experimentation that you're undertaking?
So I think the best example to address that question is our work in type 1 interferon. So I presented some of this earlier, but for those not familiar with it, type 1 interferon really crucial for antiviral immune responses, but also can be perturbed and lead to many autoimmune diseases, also a crucial role in cancer. And for many years, we weren't able to detect the protein because it's such a powerful cytokine. It's at very low levels in bodily fluids, so we just couldn't detect it. And people used proxy readouts of interferon-stimulated gene expression or functional activity. And with the combination of Simoa and some novel high-affinity autoantibodies isolated from APZ patients, we've really pushed the sensitivity down and can now quantify the protein. And we're learning a lot with that, in that we can now better stratify patient populations. We can rule in or rule out the role of interferon alpha. So we have some interesting infectious disease cohorts where there's a strong type 1 interferon signature, but we don't see the protein. And people just presumed it was there because of the gene expression. And so that leads to the second part of your question. If we could go further, there's another type 1 interferon, which is interferon beta. And the antibodies are pretty poor for that. We have an assay. And, when you plot them, we can see the alpha levels all the way down to healthy. It's, 0 point something femtograms per ml. But beta, we're at about 100 to 1,000 femtograms. So we get some of the cohort, and we know the rest are just underneath. So pushing that sensitivity with the beta assay, we'd really be able to define whether these diseases are driven by alpha or beta, and that's a huge question in the field. And if you knew that, you could target one of those with neutralizing antibodies, and you could really potentially treat a lot of diseases just by having that information of whether it's alpha or beta.
So in a way, you're saying that the actual target could be better defined for the drugs if you can get to that next level of sensitivity, alpha versus beta, and that could lead then to some breakthroughs in treatment.
I think so, right? Because a lot of people are targeting the receptor, because that way you knock out both. But the downside of that is you're clearly inhibiting a lot of important immune responses. So if you could selectively target which one of those cytokines, you would have a lot more power to really better stratify patients and have more stratified treatments.
And maybe improve toxicity then in a way too, because you could maybe eliminate some of the downside that you're putting onto the immune system today by getting them a little bit more specific.
Absolutely.
Excellent. Kai?
Yes, if I try to transfer the question a little bit to our portfolio, I would say maybe 10 to 20% of the project would have some benefit by more sensitive analysis. In particular, some biomarkers are close to the limit of quantification. In particular, if we leave plasma and serum and look for other alternative specimen, matrix types, CSF was already mentioned, but also for eye diseases, aqueous humor, vitreous humor, matrices of interest, and here monitoring of some protein biomarker with higher sensitivity might be a really step forward.
So where do you think you are with macular degeneration and the ability for drugs to have good response? Do you see scenarios where you can get yourself to 80 to 90% response with efficacy, or is that out of the question?
Yeah, this is complex, but this is possible, and at least we have some projects in the portfolio which are close to this type of efficacy, at least for a specific group of patients. And here we come to the field of companion diagnostics, patient stratification. And here, once again, a better understanding of PK-PD relationships, better understanding of signal pathways, also to make learning from ongoing clinical studies for one project to get ideas for new targets is an important side effect of monitoring biomarker, maybe not as a decision maker for the project, but as an exploratory biomarker to get a broader understanding and to get inspiration for new much more potential targets for treatment by biologics.
Excellent, excellent.
Yeah, I was just talking to Kai before we came up to the stage, and we were discussing ultrasensitive methods. We're currently participating in an oncology trial where the drug is so potent that it has to be dosed at very low levels, and those levels are so low that the standard immunoassay platforms cannot detect it. So we were really stuck to a very high sensitive platform, and using this platform enabled us to make proper PK data. So even for pharmacokinetics, increased sensitivity can be very useful.
So is that an antibody drug or a protein drug?
It's a biological, yeah.
Interesting. Yeah, so that whole area of PK-PD is something that for particularly highly potent drugs seems to be a field that is becoming, more and more challenged by the lack of sensitivity. So, absolutely, excellent. Rudy.
Yes, new technologies with a much higher sensitivity would allow to identify new biomarkers or proteins which we are not able to detect now. But I think this is not the main important point. For me, an important point is that the currently measured cytokines, chemokines, at a very low level, especially in healthy volunteers. So most of the time we compare some detectable levels in patients with almost zero in healthy volunteers. If we can go down with the sensitivity, we would be in a much better range, and we could detect and quantify these molecules much better and much more accurate. So it opens the field to new biomarker, but also improves the currently known one.
So I'm intrigued that you do both breast cancer and colon cancer. I'm wondering if you could make some comments regarding biopsies of tissue versus maybe what is in the blood as it relates to, one time we had a pretty big discussion and talk from Mass General that was doing a lot of circulating tumor cell work with our technologies, and their view was the tumor signatures and profiles that they were finding was very different than the cancers they were finding in the blood that were moving around, and they felt that a lot of the investigation of what drug would be actionable was based on what they were finding in the tumor, but yet what they really wanted to kill had different signatures because it was in the blood circulating. And I just was wondering if you've had any evidence of differences between what you might find in tumor signatures versus what you might find in liquid biopsy, and is there any potential avenue of opportunity when you compare and contrast?
Yeah, when you look at biopsy are very nice to identify mutations and resistance genes and so on. So this is fine to do genetics, to describe the tumor on this base. But when you look at the interplay with the immune system, there it's completely different depending on where you take the biopsy. So it's locally different, and also those cells which circulate in the body are different to the main population in the tumor itself. But the circulating ones are the dangerous ones because they can then form the metastasis, and that's what you want to prevent most of the time.
So in effect, if we continued the practice of using primary tumors for trying to pick the right drug regimen, you might end up in a little bit of a cascade of not really catching up to what the right signatures are that you're trying to kill.
Yes, the tumor, it's well known that the tumor is heterogeneous, And so about 70 to 80% of the cells within a tumor or in a surgical specimen are from one subtype, and there are some minor subtypes. And if you get the right drug to kill this 70-80%, the others survive. And this is a problem. And also those circulating ones might not really reflect the major population, but a minor one which is able to dissociate and go to the blood.
Yeah, it seems like an incredibly important field for sensitivity to kind of get after. So, Thomas.
Just to add on your comments, in the field of oncology, therapeutic treatment regimes are nowadays well established. Because if you have, let's say, the routine diagnostic, especially in the field of breast cancer, the clinician knows for 95% of the patients how to treat this breast cancer according to the current stage. The drawback is definitely if there is a remission and you think about second and third line treatment. And for those treatment regimes, we still have a lot of uncertainties. There is a lot of efforts going on with patient-derived 3D organoids, which you can culture ex vivo and then identify appropriate treatment regimes. And there is also a lot of individualized responses visible. So it's not as easy that we can do with ultrasensitive assays right from the beginning the right biomarker treatment, with the right biomarker measurement have a better treatment options. If there is 90% of the tumor, this 90% has to kill it anyway.
It's very interesting work to kind of think through that whole aspect. I was also curious if the panel has any views around protein versus DNA and RNA, any perspectives that you might have around the relative role in your research and what maybe is missing for you as it relates to either one of those molecular forms and what you're ultimately trying to strive for. So I was curious if you had any views on molecular versus protein and the relative role that they're playing in your research.
Yes, I mean, it's complementary, I would say. The DNA allows you really to describe the tumor and the mutation profile of this tumor. But when you go for the immune response, then the regulation is more on a level of the proteins. And all these cytokines and chemokines, it's always a question of the balance between these in their final form, in their final modified form, and not in the mRNA expression. This is mainly because this protein reaction or this post-translational modification of the proteins can occur very quickly. So the immune system is able to adapt very quickly. And so it is very important to have a complementary protein analysis to the DNA analysis. Both are necessary.
So the genetic analysis is, from a technical perspective, so much more advanced. We always say genes are easy. We have we have no PCR equivalent within the world of proteins. To analyze proteins, you need antibodies, and you need really good antibodies. With poor antibodies, even a quanteric system is not helpful at all. So to get beyond the static view of an DNA-based analytics, you can have of course, transcriptome data sets, but post-translational modification, like intra- and extracellular signaling, is mediated by proteins. Therefore, we have to look into proteins.
I have to admit I had the same in mind. This complementary approach is absolutely right and never act in any way dogmatic. But if we ask ourselves what kind of improvement we can achieve, in particular for protein biomarker, I would also like to go back to a statement by Thomas before, if I understood him correctly. If we can monitor a biomarker ultra-sensitive, but this biomarker is of no or less relevance, we have to look for other ones. And this brings us to the need for multiplex technologies, in particular in the exploratory way, in the early way, to be broad enough in the beginning of a project to understand relationships between treatment and biomarkers. And here, from the technology perspective, there's also still space for improvement.
So multiplexing is key in your mind for the protein exploration, particularly in the fields that most of the five of you are pursuing. Is it the economics? Is it the sample preservation? Is it having the same answer from the same sample? Like, what is it about multiplexing that causes you to feel like that's so important?
Maybe at first it's respect for the task, for Earth, for nature. We human beings are so complex and we understand a lot, but there's still a lot to learn. And we try to treat cancer for decades. In some cases we are successful. Maybe this is part of the answer. There is, from the biological point of view, still a need to get a better understanding on not only on the biomarker, a protein, maybe also on the subtypes of this biomarker. Maybe it's much more interesting to monitor an isoform of a specific protein or to monitor a modification. And this brings us to another important point for all technology and service and assay providers, the clear understanding what is measured. This is absolutely key, and this is important, I think, for both for academia as well as for us in the industry to really understand what is measured, because this is often also the reason why maybe the same parameter analyzed by different assays, by different technologies, give completely different data.
Mm-hmm. Yeah, I think that's an area of complexity that I don't see getting solved in the short term, but I think you're spot on. And this area of multiplexing, the way you describe subtypes, I also, before you comment, Darragh, I'm curious about homebrew. You know, homebrew is something that we've put a lot of work into, and even with the new technology that we're launching, SPX, we're creating a homebrew strategy, because it does seem like scientists like to come up with their own multiplex based on what their specific strategy is. And I'm curious if the multiplex homebrew is something that you would see a lot of versatility for, given that your subtypes might be different than the next guy's subtypes, and you need to customize your work, I would think.
Yes, I think so. And I see two big advantages. The option, an open system which allows homegrown assays, gives also our academic colleagues much more chances to develop and to optimize assays, maybe with their own tools. And also for us in big pharma, it helps a lot because for all assays, reagents are key. And any technology provider might be limited to a certain set of antibodies for capturing detection. And a company like ours, looking on several parameters, targets, have maybe a huge toolbox, maybe in some cases a better toolbox, and therefore the option to consider at least homegrown assays would result in better assays. I'm convinced.
That's excellent input. Darragh?
Yeah, I was just going to comment on the gene versus protein debate, and yeah, completely agree. It's complementary. I think you need to do both if you can, but also know the limitations of both. With the genetics now, we can do a completely unbiased approach. We can sequence the 3 billion nucleotides that are in individual cells. We still don't know everything of what they're doing, but you're doing an unbiased approach of the data and letting the data guide you. With protein, you've got to know what you're going after and target that. The downside of the genetics is that even if we can sequence everything and we know the whole code, from the current models, it's still only explaining 20 to 40% of the variability. So there's a lot of things that can go wrong from the genome code through to the phenotype, and that's why we like proteins, as you mentioned this morning. It's closer to the disease phenotype. But then you've got the other layer that was alluded to, the post-translational modifications. That's just we're only beginning to understand that, especially in immunology fields. So
I'm going to open it up to the audience, but I did want to also raise the question around comorbidity and the linkages. Like, we've thought about cancer being organ-based, and now we're moving to defining cancers with a lot of different types of signatures. It could be any place in the body, and I'm just curious, like, you know, the role of and linkages between infections and cancer, and even like neurodegeneration. It just seems to me like biomarkers might hold a promise of seeing some linkages between different diseases that today we are just at the tip of the iceberg, and someday maybe if we really understood how all that interrelated, diseases that we categorize as infectious disease versus cancer versus neurodegeneration might start to blur a little bit. I was curious if any of you had any views that could be interesting for the audience to hear as it relates to how one category of disease might be related to another category that we just don't quite understand at this point.
So I think it's a really interesting area of future research, is trying to understand these links. You know, when we work in our own silos, you only learn so much, and then you step into another field, you can learn a lot. And there's clear links between infection driving certain cancers that we know about. I'm sure there's others that we don't, but also the autoimmunity link that's really coming out of some of the cancer immunotherapy studies, that when you unlock these checkpoint blockades that are great for clearing the cancer, you're getting all kinds of autoimmune problems flaring up. So we're learning a lot, and we need to learn more. And I'm sure that they're all linked. You know, if you have a genetic predisposition to an autoimmunity or to a cancer, there's some advantage against another pathology somewhere. You know, evolution has selected that for a reason, which is why one reason we focus a lot on infection, because that's been driving a lot of the evolutionary selection. So if we understand how different populations have evolved for their infectious response, we think we can better understand the risk for autoimmunity as well.
Excellent.
And just adding to those comorbidity factors, everybody is growing older, and of course you acquire additional diseases during your lifetime. And a biomarker in the background of a lot of other issues going on within your body may be not as good as if you just look into more or less early young people with early detection of cancer. So individual baselines is well known for a variety of proteins which are discussed as biomarkers, and as long as you do not have the biological understanding what's going on, then it's sometimes useless to compile a huge amount of data sets. Cancer antigens, which are nowadays routinely measured, are only recommended to be measured during therapy, whether we see that we can see the efficacy of therapy. If you would use them as a monitor of remission, you always, whenever they come back from the clinician and get their baseline data, they are always a little bit above the threshold, so you always will induce fear. That's why it's not recommended at all to measure those cancer antigens as a prediction for remission of cancer.
So I completely agree with the view that there is an interconnection between the different diseases, and that especially the immune system is known now to interfere or to have a crosstalk with the cancer cells, but also it plays a role in the development of Alzheimer's disease. It has also an inflammatory perspective. But this makes it also more difficult if you want to find diagnostic biomarkers, so it's even a bigger mess than we thought before. But in the clinical the routine, most of the time we have already diagnosed patients, and we have the question how to deal with them. So then it's very nice to have also some experiences from other types of diseases, because this describes comorbidities or also how the patient is responding to the therapy.
Excellent. So, we had a good lunch. Is everybody still alive and well out there? So, how about some questions from the group, please?
Microphone is coming.
I'm Brandslund from Denmark. The question about sensitivity. As a clinical chemist, clinical biochemist, and a medical doctor, I would prefer if I have a test that I am able not only to measure increased concentrations, but also normal concentrations. And now it comes below normal concentrations. So any test you have, you should be able to measure below quite normal concentrations, because if you cannot do that, you cannot find out whether a lack of that protein matters in disease. So that's the demand, to be able to measure below normal concentrations. It's not about picograms, femtograms, whatever. It's below concentrations. So you can say this is very much below the normal range. This is important. It wasn't believed important for thyroid-stimulating hormone when we started measuring that, but suddenly we could measure in the second generation in the normal range, third generation below the normal range, and suddenly we had a test for thyrotoxicosis. So it's the same with complement C3D, which I have developed. If you can measure 0 levels or close to 0 levels or at least below the normal range, you can actually screen for complement deficiencies. And if we couldn't have done that, we could not have found out how myelomatosis inhibits the complement system. So you need to be able to measure below, whatever the concentration is.
But may I comment, but not for everything, because when we started using Seymour technology, we were measuring procalcitonin because we had the assay established, we had unlimited amount of antibodies, and then we went to our clinician. Look, we can measure procalcitonin baseline from young, healthy volunteers. And he was looking at us. So what? Nobody cares. We just are interested if procalcitonin is getting up. So again, if we understand the biological relevance, then it's easy to define what we need to measure. And of course, I totally agree, for some of the analytes, we have to measure downregulation to learn more about them.
The thing that intrigues me about your point as well is that we've been seeing such incredible range in the baselines between individuals of some of these markers, particularly the cytokines, sometimes 2-3 magnitudes of difference, the quiet baseline differences. So this whole concept of knowing your baselines as an individual someday, I think, is going to be incredibly predictive and helpful for how you live your life. Because I think everyone's baselines, based on whatever it is that created those baselines, there can be a big range.
Yeah, one of the points that I think is important is that you try to get your information out of your trial as early as possible. So I know even in phase 1 you start in healthy volunteers, but deep down in everybody's heart, of course, safety of the volunteer is priority number 1, should be. But deep down in our hearts, we want to know if the drug is doing something, right? So if we talk about cytokines, for instance, measuring cytokines in a healthy volunteer in the circulation is already not that easy. It's even difficult to measure cytokines in patients unless you go to the inflamed areas. So, but if, for instance, you're dosing an anti-TNF antibody, what would be your primary PD marker? That would be a drop in TNF in your serum. Well, if you have a sensitive platform, then you can analyze it. By all means, try to squeeze that information out as early as possible.
So that's a key point, is the speed of knowledge is paramount, and sensitivity can absolutely play a major role in accelerating the knowledge that you can pull off of the markers.
Just wanted to ask a question. Talking about potential target for very sensitive platforms like Simoa, for example, and you mentioned DNA before in terms of genomic studies. What do you think about circulating free DNA in oncology? Does it have a future in the diagnostics, prognostics? It holds some promises in the neurodegeneration field, which I do belong to. But
Yes, it's a very good question. The circulating free tumor DNA is a very interesting field. We also did some studies, or published this year a study on circulating tumor DNA in the response to therapy in colorectal cancer with liver metastasis, and we could really show that there is a correlation between the tumor volume and the decrease in the tumor volume, and even in the remission with the circulating tumor DNA. So this is a very interesting thing and also very sensitive, because we have this PCR. In that case, we looked for methylated DNA. And yes, I think this is also interesting, but it's not a platform. It looks at the tumor itself, but it says nothing about the interplay with the immune system.
So we had a major debate at one of our Powering Precision Health summits over this point. And Johns Hopkins is pretty significant in this field of free DNA, and we have some other scientists that felt that most of those cells are dead that are shedding the free DNA, and that's not what you're most interested in. And so it became a pretty big debate and a lot of provocation around this. I don't know if Mark Hrusovsky's in the group here. Mark, it might be good for you to just let everybody know that we can do DNA and RNA at an exquisite level of sensitivity without PCR. It's something that we've not really pursued, even though there's a lot of homebrew going on today on the SRX particularly. But it might be good if you can just give them a little description of that, to the extent that that can play a role in some of your research.
Yeah, just the actual same core technology that enables the sensitivity for a protein assay, doing a bead in a femtoliter size well and constraining the single signal, can be applied to DNA and RNA through a hybridization-based approach without amplification. So we can get to PCR level sensitivity with non-amplification, and hence we haven't really developed it out as a company yet, but that technological capability is there. So.
And Destina is somebody that we've done some collaboration with, I think, against microRNA 122 for liver toxicity. And so there's been some assays developed, but in general, it's still an area that there's several publications of folks
Correct.
that have done it.
Yeah, in particular, microRNAs, small microRNAs are hard to PCR amplify, so the technology is very adept at detecting ultra-low quantities of small microRNAs. And Destina has developed assays for microRNA that's related to liver toxicity, and we've been working with a few other customers in early R&D around specific microRNA detection. So if you're interested in that, please talk to me or anybody else from Quanterix.
And in the landscape of infectious disease, it seems like Merck and others have presented at previous PPHs around wanting to look at the DNA, RNA, and protein all in the same sample to kind of understand that cascade. And so that's some of the places where we're starting to see people diving in on this concept of utilizing the Simoa for DNA and RNA. But it's not been our primary focus, and we don't have a lot of resources dedicated to it. But to the extent that you guys start to get really excited about that, we can change our resources depending on what you see.
Yes, I would encourage you to further investigate this field. Maybe one short question to Mark. Does it matter, or does matrix type matter? Do you see differences between tissue analysis, plasma samples, urine?
Well, again, there hasn't been a ton of work done yet, but some of the early work is actually pretty exciting, because it looks like there's a protocol where you can keep the samples in a native state, no freezing, and you can directly detect the microRNAs anyway in a variety of different sample types pretty accurately. So I can't say for sure that a particular sample type is going to work or not going to work, but I would say we've tried a number of different blood, serum, plasma, saliva, tears, and been able to really get good signal from microRNAs.
So Martha, I think you might need to be talking to Kai about an SRX, because I think there's some interest here is what I'm sensing. Other questions from the audience around any of the topics that we've been discussing up here? Everyone's awake out there. This is awesome.
I guess this is for any of the panelists, but are you seeing any association with the microbiome and oncology drug responses?
This is a very interesting question, also a complicated question, because we became more and more aware that that bugs can be found in parts of the body which we thought that they are sterile. So we have now available very sensitive methods, DNA-based method, to amplify these ribosomal DNA structures from microbiota and find bacteria within the tumor itself, bacteria in the mucosa of the colon, bacteria also in the mammary gland in healthy patient without any sign of inflammation. So there is, of course, something coming or there's something occurring there what we do not understand right now, because obviously the immune system is tolerating very few bacteria in otherwise or assumed sterile tissue. There is no response, only when they start to grow. In the oncology field, especially in the colon, where we have a lot of bacteria in the lumen of the colon, of course, we do not know how much the bacteria are interfering with the immune response. But one important point is that intestinal immunity is very immunosuppressive, because it has to live with all these bacterial compounds, which is in the daily life going through the mucus and to the mucosa. And this is also the complexity, also that makes a lot of difficulties to understand the response to the tumor, because the immune system is already immunosuppressive and the tumor is well tolerated there.
I can just comment. There's been a few high-profile papers that I guess you're aware of showing an association between microbiome and cancer immunotherapy responses. It's not something we particularly work on, but we're more focused on understanding how the microbiome controls healthy immune responses. So we have 1,000 healthy donor cohort where we're exploring that, and it's still ongoing. But I can tell you that we've already done tried to do the genetic associations, at least at the 16S level, and we don't see any genetic associations. So there was a couple of early high-profile papers in Nature Genetics showing these genetic associations, but very few people can replicate them. So the whole microbiome area is very exciting, but it's a bit of a wild west at the moment, I think. So the sort of early studies that come out, I think we need to be very careful with the conclusions, because the microbiome has been shown to be associated with everything at this stage.
Great. So Darragh, are you eating yogurt each morning? Is that something you would recommend?
I mean, I live in France, so people eat yogurt every day. Okay, maybe that's why our long life is pretty good.
Just check. And the other thing too, I didn't show it in this presentation, but women around the world live 7 years longer than men, and it's just amazing to me, like the studies that I've seen around some of the antibodies that maybe even through pregnancy and children and crossing over of the barriers. So that's like another whole field, and longevity. It's the whole research of longevity has been intriguing, and there seems to be a lot more interest in some of the biomarkers in that whole landscape. Please, Mark.
Great. Thanks for the presentation. I guess a two-part question. One is there seems to be some moderate degree of debate in the United States whether or not DNA can shed in the bloodstream for colorectal cancer. That's my first question. The second one is, there's a lot of money flowing into developing an early stage blood or saliva-based test for liquid biopsies across cancers, and I guess my question is, One example is Grail, formed out of Illumina, and they've invested over $1 billion, but they seem to, their goal is to find, a blood-based test for asymptomatic patients. And I'd be curious to hear your thoughts about the feasibility of developing a test with such ultra-high sensitivity, so deep that it could detect, cancer in asymptomatic. It seems to be a riddle that has a lot of skepticism. And so I guess there's a whole other wave of companies going after individuals that have a high genetic risk of cancer. There's 20 million Americans that have a higher predisposition for developing cancer. Maybe it's family related. So I'd love to hear your thoughts on the likelihood of some of these approaches working for early stage cancer detection.
And before this distinguished panel answers this, Mark is with Canaccord, and so worldwide markets could change tomorrow as a result of your answers. So good luck.
So regarding the circulating free tumor DNA, I can say that we can find this free tumor DNA in patients with liver metastasis or with a peritoneal metastasis, but colorectal cancer patients with stage 2 or stage 3, I really don't believe that you can find it. There are several studies showing, but I think it's not so good reproducible. But with metastasis, yes, I really am convinced that it's really good for us to follow the development of the cancer. Then regarding your second question, the very sensitive analysis of patients with asymptomatic patients. To be honest, I do not really believe that we will find a diagnostic biomarker which is so much better than the current diagnosis. I think the biomarker should focus on later stages, the development of the diseases when we know already, when we have the diagnosis, because most of the proteins which go up in cancer patients, when you compare cancer patients with healthy volunteers, you can find the same biomarkers in inflammatory bowel disease or in the similar biomarkers in autoimmune diseases. So the specificity is the big problem. It's not the sensitivity, but the specificity for specific disease.
May I just add to this? If you want to answer your question about, let's say, a screening approach, you should go back to the last 20 years of cancer-related biomarker research. 20 years ago, big pharma companies, Jan von Ostrom, Novartis, your colleague Hanno Langen at Roche, and Dennis Hochstrasser, in the proteomics field, cancer biomarker was on a really high level, and a lot of cancer biomarker candidates were identified. Samir Hanash was looking into cancer autoantibodies, and he was coming up with great data sets that post-translational modifications of tumor antigens are key to visualize cancer half a year before clinical manifestation. There are bits and pieces around, but no generic approach could be placed into the market because of the clinical specificity you would need. You have to avoid false positive calls. Otherwise, everybody gets fear, and of course, that's good for the test business, because every second month they run the same test again and again. But it's not, the overall benefit is really poor, and huge projects were stopped after a decade of efforts. I would be more than willing to tell some more details on this topic.
I would also comment that we've spent a lot of time with Grail, Chief Medical Officer, and learning, and I think they initially were very much a next generation sequencing focused, and they are trying to become more agnostic so that they can go after a broader swath of technologies to maybe get at what I would consider to be a better opportunity for the answer. Freenome is another company that does now have a Simoa technology, and they're trying to link up the genes, the DNA, the RNA, and the protein, looking for signatures for different cohorts that actually do have cancer, and then looking for some of the recurrence syndrome, which might be a cleaner place to move into this. I think you're hearing a little bit of that from this panel. But I do think there's a lot of different, there's a lot of hype right now in that landscape of liquid biopsy, and we're trying to carefully measure what we can pick up in these summits around what platforms will ultimately provide the kind of insights and perspectives. And so I do think we're feeling like what we're learning is that DNA, RNA, and protein, all in some level of collective AI algorithm, might give you the best personalized understanding of some of that pathology, but it still seems very early. And I would encourage that we have these discussions, during the break as well. Great learning here. I also wanted to announce before, I think we're going to break now or head into the next sessions where we go into neurology here and oncology upstairs. Barry is going to start off upstairs. I think the agenda showed him going this morning, but the discussions were so productive in the sessions that Barry's going to start off. So that's going to be upstairs. Thank you very much. And